Welcome! In this lesson, we build an intuitive picture for how smooth endoplasmic reticulum forms branched tubular networks throughout the cell. We will combine simple geometric arguments with the action of specialized proteins to understand why tubes form, how branches are stabilized, and how these networks interface with the plasma membrane. First, a global view. Inside the cell, smooth tubules weave a network that can reach out toward the plasma membrane. At specific spots, these tubules touch the plasma membrane to locally trigger processes, for example at axon tips or adhesion domains, where signaling can be amplified. A generic driving force is geometric. The endoplasmic reticulum aims to maximize its area relative to its internal volume, helping maintain osmotic homeostasis. For three simple shapes, the area to volume ratios scale like sphere 1 over radius, tube 1 over tube radius, and disc 1 over thickness. Since tube radii and cisterna thickness are comparable, both boost area efficiently, but tubes can extend far through the cell without blocking traffic. The lumen is continuous throughout the cell and connects with the double membrane of the nuclear envelope. Critically, calcium is high inside the lumen and low in the cytoplasm, so geometry also shapes calcium signaling. What regulates the complex topology? Reticulon family proteins. They have hydrophobic alpha helical segments that insert like wedges into one leaflet, sensing and sculpting curvature. They prefer highly curved edges, like tubules and rims of cisternae, and they cluster at junctions to stabilize branches. Reticulons work with partners. REP proteins link tubules to microtubules, the cell's transport tracks, and together with spastin, they can sever microtubules during cell division. If microtubules are disassembled, the network quickly collapses. Another key player is atlastin, a fusion GT passe that stitches tubules together by homofusion. Two atlastins on opposing membranes bind and pull the bilayers into a diaphragm, then fuse. tubules at the periphery can make close contacts with membrane signaling hubs. One example, a phosphatase anchored in the called PTP1B dephosphorylates and activates SRC kinases at nearby plasma membrane domains. How hard is it to bend membranes into tubes? Not very. Making a tube of about 20 nanometer diameter from a membrane of bending modulus around 10 kBt costs only a few hundredths of kBt per lipid. That small cost means generic factors like osmotic pressure and spontaneous curvature can drive tube formation, while reticulons position themselves at curved regions and build functional machines. Could tubes extend all the way to synapses and control calcium at axon tips? Long unbroken tubes are unlikely to stay intact over centimeters. More plausibly, tubules break and refuse repeatedly, assisted by atlastin, enabling stop-and-go transport while still supporting fast calcium waves on the order of 30 micrometers per second. To summarize, 1. Maximizing area to volume favors tubes and cisternae, with tubes offering far reach. 2. Reticulons sense and sculpt curvature, stabilize branches, and assemble machines with partners like our EEP, spastin, and atlastin. 3. Geometry underlies calcium signaling and local control at plasma membrane contacts. Together, generic physics and specialized proteins create adaptable networks.